In this episode of Florida Field Journal, Donna Day, a volunteer at the Carlton Reserve, is examining some dead giant air plants for evidence of infestation by the Mexican Bromeliad Weevil. This is the Florida Field Journal.
Center lifting out. You can see the holes really well on that one too. There's a pupa in here. I'm going to uncover it. And without killing it. Oh, no, it's still in larval form. But that's the part that does so much eating. There's probably another one here. It'd be fun if it was a really clear pupa.
Can you feel it wiggling when you hold it?
You feel there's something there, yeah. Oh, we got a weevil. I don't want it to fly away, but I want you to get a shot. This is characteristic. It's either going to be yellow or orange, and I'm going to be ready to put him in this vial.
It's only moving.
Yeah.
Very cool.
Isn't that amazing? So I'm going to put him in here. We'll keep eye on him. We'll give a little bit more plant material, because I think one of the keys to keeping him alive is
Feeding them.
yeah, exactly.
Isn't that
Keeping
the
him
truth
moist
with all of
and-
us?
Yeah, food and water.
Was that emerging?
Yes. 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 And whoops. He's going to wander around in here. Oh my goodness. Well, I'm really happy that we got him, because that's one less that can be out there killing plants. It's just strange that. Yeah.
Got holes in it.
It looks like there's still some seed here. So what we want to do is take care of the spike first.
Yeah.
Yeah, because we can put the spike up in the tree and let it do its thing.
And these don't die after they spike.
No, they don't. So that was a big clue.
Yeah.
And it just didn't. After a while, you look at these and they don't look right, you know.
Yeah.
I've never seen one in a fasciculata before, so this is interesting. And the native weevil doesn't kill the plant. It distresses it, but doesn't kill it. Oh. There's a weevil. There's a weevil. Is it alive? I think it's dead. That's wonderful. What'd you do to it, Shirley?
What would cause its death?
I don't know.
Too much to eat.
I don't know.
Dead?
Yeah. But it's a really good specimen, so you can see. Oh my God.
Yeah.
They're quite old,
Yeah.
so they're smaller back there. But that's interesting that it's in that big of a...
It's in fasciculata,
Isn't that weird that it's
and
there?
it's in the second.
Is the white around it, is that typical?
Oh, we've never seen it? No, that's not.
Usually, no, that's not. That's there. But did you notice
Where did it
on
looks like
several
a
of the dying
fungus.
ones
Yeah.
that there was the white? around the base? Because
Yeah.
I hadn't seen that before.
Something struck back.
Divine intervention.
The Mexican Bromeliad Weevil attacks most of our native bromeliad species, but is it as deadly to the others as it is to the giant air plant, the Tillandsia utriculata? I caught up with Dr. Teresa Cooper at the Loxahatchee National Wildlife Refuge to find out more.
My name is Dr. Teresa Cooper. I'm with the University of Florida at the Indian River Research and Education Center in Fort Pierce. We work with invasive insects there using primarily biological control. And the main project that I'm working on is the invasive bromeliad eating weevil, Metamacius calazona, or the Mexican Bromeliad Weevil. And it's here in Florida, and it's destroying our native bromeliads. 12 species of our native bromeliads are affected by this weevil. And our efforts is to try to find a way to control the weevil and to conserve the plants in Florida. These are root balls that were once living plants, Tillandsia fasciculata. And when you first came through the forest, these plants were growing all in the canopy. The weevil came along, killed the plants, and the plants fell out of the canopy, and this is what you have remaining on these trees are the root balls. So you can imagine that where these root balls are now, once there were a whole lot of plants.
Like that one out there.
Like that one in the back. You can see it has the inflorescence coming out. That's Tillandsia fasciculata, yes.
So that one still is alive and viable.
It is still alive and viable, but still in danger of being infested by the weevil because the weevil is still here in the forest. So there is a weevil that came from Mexico into Florida. This weevil is a specialist of bromeliads. That's all it eats. And in Florida, we have 16 native species of bromeliads, and 12 of these species are being attacked by the weevil. One of the plants, the giant air plant, is being particularly harmed by this weevil and actually stands a chance of being extirpated in Florida because of the weevil. The bromeliads are epiphytic. They live up in the canopies of the forests, and they actually create another level of habitat up in the canopy where animals live and find refuge. And they use the bromeliads for building nests and for hunting grounds. And with the giant air plant, what you actually have is the plant holds water in the axils of its leaves. So you have a giant population of these plants up in the canopy, and it's actually like having a swamp up in the canopy, little ponds all over the place. And they support aquatic ecosystems. It's called phytotomata. And actually there are species of insects that live in the water in this plant in Florida that are found nowhere else in the world. So if we lose the bromeliad, we lose these associated arthropods, and it's not just the loss of the plants themselves, but the insects that they support. And the change in the canopy, the loss of the habitat up there, the loss of the water up there, and the change in the nutrient flow, because the nutrients that were being held by those plants are no longer being held by those plants, but just being released to the canopy floor. So it changes the structure of the biodiversity, and it also changes the nutrient flow in the forest. They're very ecologically important to the canopies in Florida, part of a unique part of our flora and fauna. So it would be a devastating loss at many levels, loss in biodiversity, loss in functions happening in the forest. And it's so far-reaching, and we can't even fully describe everything that's going to happen, all the changes that will happen.
We certainly don't want to pump pesticides into these pristine forests. What are the alternatives?
Yes, you're correct. We certainly don't want to use insecticides out in the natural areas in Florida. Not only would it be dangerous, but it would also it just wouldn't work. Initially, the means that we tried to control the weevil was using a classical biological control. And this is where you go back to the place where the pest originally came from, and you look for a specialist natural enemy that will attack the target insect. You bring that enemy to where the pest is newly located. You study it for many years to make sure that it is specific to that host, and then you release it. And the hope is that this natural enemy will become established in the forest and will attack the target insect and keep it under control. It won't eradicate it, but it will decrease the number so that it's no longer causing the problems that it's causing. In this case, we have the weevil that is causing the problem, and we went back to Central America, which is where the weevil came from, and we searched for a specialist natural enemy, and we found one candidate. It's a tachinid fly. It's called franki fly. Its scientific name is Lixad montia franki, and it's about the size of a housefly, and its maggots will actually attack the weevil larva. It'll find the weevil larva in an infested bromeliad, and it will use its little mouth hook to cut a hole in the skin of the weevil larva, and it'll go down inside of the weevil larva where it will eat it up from the inside out while the weevil is still alive and functioning. And about a week or so later, the weevil larva will die, and the maggot will come out of the weevil larva and pupate and then become an adult, and the cycle will go around and around. We studied this fly for many, many years under quarantine conditions, first in Central America, then in Florida under quarantine conditions, and we determined that it was specific to bromeliad-eating weevils. And then we received permission to release the fly, and we actually made several releases in Florida, including here at Loxahatchee. And after we released the fly, we did post-release monitoring to see if the fly had become established and if it had been having any kind of effect on the weevil populations. Well, we only got one fly back from the post-release monitoring. That was after the very first release, and we were pretty excited about that because we thought, you know, we were going to have some good luck with this fly. But after several years and several more releases and post-release monitoring, we haven't had another fly captured, and we haven't seen any evidence of it having any effect on the weevil populations in the areas where we released the fly. So it's possible that the fly may be out there, but even if it is, it's not having an effect on the weevil population, and we're starting to look at alternative methods to try to save these bromeliads in Florida.
So what other options do you think there are?
We have two new approaches that we're taking right now. One is looking at host plant resistance. If you go to Belize, there's a population of the giant air plant growing there. It's the same species that we have growing here. And in Florida, this is the species that's getting hit the hardest by the weevil. But in Belize, same species, but it's living with the same bromeliad-eating weevil, the Mexican Bromeliad Weevil, but it's a healthy population of this of the giant air plant. It makes sense because in Central America, that variety of the species has been co-evolving with the weevil for many millions of years, and so it has apparently some inherent resistance to the weevil. In Florida, we're kind of on the outer boundaries of the range of this plant, and here it's been isolated from the weevil for millions of years, and so it doesn't have that resistance. It hasn't had the necessity to get that resistance. So our question is, why is the variety of this plant in Central America resistant to the weevil? And can we take that resistance and hybridize it into our variety of this giant air plant, and therefore maybe have a population in our forest in Florida that are more resistant to the weevil? So that's one approach that we're taking. We've done some laboratory research, and we have looked at the growth and development of the weevil on these two varieties of the giant air plant. And there is a distinct difference in the growth rate and the preference for oviposition and survival on these two species. We still have some more research that we have to do in the laboratory, but soon hopefully we can start looking at how we can get this resistance into our form of the plant. The resistance, it seems to be primarily that there's just less available nutrition on the Belize form of the plant, and less nutrition available per area for the plant, and that seems to be the primary reason why you get slower growth rate on the ones in Central America. But we still have a lot of research that we have to do on that before we can get it off of the ground and running. The other approach that we're taking is we're looking at doing active conservation with the species. We're starting with the giant air plant because it's the one, again, that's being hit the hardest by the weevil, and because the area where I'm starting the pilot project for this is at the Enchanted Forest Sanctuary up in Titusville. We have historical data there dealing with the bromeliad population there, the giant air plant population. So we have that historical background to work with. And because the population was primarily the giant air plant, that's what we're dealing with there. The idea of this project is to get many different people involved throughout South and Central Florida, primarily land managers of natural areas like here at Loxahatchee. We're working with people out at the Carlton Reserve on the West Coast, Lemon Bay Park. I plan to do this at Myakka River State Park, and just to get as many people involved as we can.
We're actually in a nursery area, which is a very exciting part of what we're doing, because if we can keep these young plants alive, they don't attract the weevil when they're very small, and perhaps by the time that they're adult and ready to spike, there will have been something found, maybe a fungus, maybe some kind of biological control.
In March of 2014, we rescued about 140 plants. 70 of these giant air plants went to Naples and 70 to Selby. And in November of 2014, we replaced 16 of them that had spikes. That's all a permit required, was whenever they make their spike, we bring them back. So it's an ongoing process, but it wouldn't happen without collaboration. So it's a great project.
I was with Teresa at Loxahatchee. That place is devastating.
Yeah.
The fasciculata is just barely hanging on.
We're going to need to do more of this with Botanical Gardens' help to repopulate, if we can get some handle on the weevil.
Yeah.
It's our only hope at this point, is just to take some of them and keep them safe.
If we can get lots of individual landowners throughout Central and South Florida to monitor and protect gardens in their natural areas, then we can keep this plant in Florida, we can keep the biodiversity, and most importantly, we can get the seed production back up. One of the biggest things that the weevil has caused, one of the reasons why it's so destructive, is because it's killing these plants when they're very large, just before they release seed. And with the giant air plant, it can take up to 10 or 20 years for it to go from seed to seed production. So you got a plant that's maybe been out there for 15 years, finally getting ready to put out seed, and then just before it happens, the weevil kills it. So seed production has been hugely diminished. And because the seeds are not viable for very long, if they don't germinate that season, they're gone. We have no seed bank out there. So we have to find a way that we can protect these plants, keep the seed production going, and meanwhile continue to look for a way to control the weevil. We've searched and searched and searched for other biological control agents that are specialists of the weevil, and we haven't found any. So what we're doing now with these gardens, what we need a way to to control the weevil, to protect these plants. We don't want to use insecticides, again, because they're in natural areas, but there are other alternatives that we can use. There are such things as biopesticides, like neem oil, which is relatively safe, and it is used to control many different insect species. Just starting to do the research in the laboratory, so I really can't give you any effect on the outcome yet, but it's a good start in looking at a means to control the weevil within these protected gardens. And other things that we can look at are like using augmentative biological control. This is where you take an organism that already exists in the area where you're trying to control the pest, and this organism will kill the pest, but they don't usually come into a lot of high contact in the natural world, so you don't get that happening. So what you do is you go and gather a lot of them up and you put them on the organism that you're trying to kill. A classic example of this is someone who's growing tomatoes and they have aphids on their tomatoes. So they go out and get a bunch of lady beetle larvae and they put them on their plants, and the lady beetle larvae kill the aphids. And after the season is gone, you know, the lady beetles dissipate back into the environment, so it's a safe way to do a control. And there are a few options that we have that we can use. There are entomopathogenic, this meaning they kill insects. There are fungi that are used for that, specialists on insects. And then there are also like some nematodes that are used for that. But again, just starting the research in the laboratory. And even though these are methods that are ecologically safe, sound, they do still have an impact on the environment, so we cannot just go out into the natural areas and start spraying stuff and putting stuff around. Can't do that. You have to study it in the laboratory, number one, to know that it's going to have an effect. If it's not going to have an effect, then there's no reason to do it. And then number two, you don't want to use more than you have to use. And you have to know the timetable at which way you have to reapply these methods of control. It's unlike classical biological control. You put it out there, it stays out there, and it works on its own. With this type of control, you have to do reapplication. So those are the things that we have to figure out in the laboratory, and then we'll start taking them into the field. And that's the two alternatives that we're working on right now.
So obviously this is an ongoing story that's going to unfold over the next several years, and hopefully there'll be some answers.
Yes, we're kind of closing one chapter and starting another chapter. So we tried really hard with the classical biological control. It's possible in another 10 years we may see some response from it, but the resources and the lack of success, we have to close that chapter for now, and we have to move on to something new. And I'm very hopeful with both of these approaches that we will be able to keep these bromeliads in our forests, keep the production going, keep the biodiversity. And the people that I work with the natural areas, the land managers, the volunteers, the people who are just interested, they love these plants, they love the natural areas, they want to keep Florida like Florida is, protect these unique parts of our flora and fauna. And when I work with them, it brings me hope that we're going to be able to find a way to resolve this problem.